Can you hear me, students? Hello? Can you hear me?
Yes, Miss.
Okay. So, be in a position to join as immediately as the site is open to you all. You're taking at least 10 minutes for joining. Isn't it? This is not good. Only 14 of you at 11:31. कॉपर सल्फेटचं पण
हो हे खाली कसं दिसतं रेडिश ते नाही यायला पाहिजे का तयार ह्याच्यामध्ये
कारण का कसं आहे की पार्टिकल्स असतील त्याच्यामध्ये थोडीफार तरी रेंज येते का
कोणी येते
म्हणजे ऍब्झॉर्बन्स ती रेंज येते
बरोबर 200 350 500 आहे 500 600 ग्राफ कसं कोणतेच की ते असं अनियमित येते
आपले पार्टिकल्स आहेत त्याच्यामध्ये
हे आता खालचं रेडिएशन घेऊन बघू.
नाही खालचं तर असं केलं डिस्टर्बच येणार असं.
ह्याच्यात पण खाली सेटअप होते.
नाही थोडंसं फिल्टर करून बघू. नाही थोडंसं फिल्टर घे, फिल्टर कमी आणि ते फिल्टर हेडचं घे कसं आहे फिल्टर करून फिल्टर करून थोडं घे. नाही तर मग याच्यामध्ये नाही चालत. तर मग हे थोडंसं होमोजेनाइज करून बघूयात. शेक करायचं, सस्पेंडेड पार्टिकल्स पण येतील असं बघायचं. आणि
फिल्टर मध्ये काय येणार बारीक येते.
फिल्टरमध्ये फिल्टर रेट जो खाली येईल त्याच्यामध्ये याच्यापेक्षा कमी प्रमाण असेल. त्याचं काही आहे का म्हणजे त्यांनी येते का रिडिंग बघूयात ना. So we have started with the unit 1. Can you hear me, please? Yes, students. Are you there? Connected? And can you see the presentation on the screen? Yes or yes? Okay. So just in the last lecture, we had introduced about the virus, its discovery, definition of the viruses, size of the viruses, observation of the viruses which can be done by electron microscope. We have also compared the sizes of viruses and I had said that the viruses, they are measured in nanometers and the range of the size, it covers a wide range of the size viruses. They have a wide range of size. And here we have compared the sizes of two extreme viruses, that is the parvovirus, which the diameter of which is 20 nanometers, and the mimicking virus isolated from amoeba, which is up to now the largest known virus, approximately 200 nanometers. So we had also seen about the viruses which are, as we have seen yesterday, they are not the cells. They do not contain the organelles except for the arenaviruses. So we have also seen that a single virus can infect a cell, use its own machinery, that is the cell's machinery, and is responsible for the reproduction of itself, having a large number of the particles at the end at the expense of the cell. That is, the cell is either, you can say, bursted, or in case of animal viruses, which are envelope viruses, they just pinch out of the cells, taking the envelope of the cells also. And some of the viruses remain in the cells for the further reproductive cycles. So the point here is a single virus infects a cell, a bacterial cell or a eukaryotic cell, and it is responsible for a crop of viruses. After the viral cycle is completed. So we have seen about the virus genes, and the virions, they contain 4 types of genomes. Yesterday we have seen this picture also, and the presentation over here of the types of the genetic material which is present in the viruses, that is the double-stranded DNA or the single-stranded DNA, double-stranded RNA or a single-stranded RNA. I've also told you some alterations in the structures of these RNA or the DNA strands, where at the length of this DNA, there may be complementary bases, and there may be again pairing between these bases wherever complementarity is there. And this results in the formation of various shapes of the viruses. Various shapes of the genetic material, whatever it possesses, either DNA or RNA. Viruses also possess segmented DNAs or RNAs. So this is about the viruses. Now we will shift further to the description part, that is the functions of the virions. That is, we have seen yesterday that the virion, it is a virus particle which is infective in nature, which is capable of infection. It is a complete structure which consists of the coat. The coat is composed of proteins, and the proteins are of different types, which are coded by the genetic material of the viruses. So, a protein coat within which the genome is present, such a particle is known as a virion. So, the function of this virion, or the protein coat rather, we will see the function of the protein coat in the virus or the virion. The protein coat is responsible for protection of the genetic material. And the intact virion, they deliver the genetic material into the cell in which it can replicate. So this is known as infection, or this is known as, you can say, attachment of a particular virus and a particular cell, eukaryotic or the bacterial cell. So viruses, the genomes that are much smaller, and there are some viruses which have a genome at a quite a larger range. Whatever the case is, viruses, uh, after passing the genomes, how does the genome function? So it is found that the genome which enters inside the cell, a bacterial cell, or it may be a eukaryotic cell, it achieves this in a number of ways. First is viruses use the host proteins. That is, the host proteins which are present, which are responsible for the replication of the genetic material, enzymes which are present, which are responsible for the duplication of the DNA or replication of the DNA or the RNA. These proteins or these enzymes, RNA polymerases or DNA polymerases, host does not, the virus does not have these enzymes. Enzymes required for each and every metabolic activity of the viruses, they are taken from the cell which it infects. So proteins is the first one. And the proteins, the question over here is, how is that the proteins or the ribosomes or the cellular machinery, the enzymes, which are of the cell itself, that is the eukaryotic cell or a prokaryotic cell, how is it that the proteins of the cell now are responsible for decoding of the genetic material of the virus? And as we all know that the viruses, the DNA of the viruses, these are foreign particles to the host cell. And we all know that such DNA, when it enters inside the host cells, it is responsible for, or it is subjected to the lysis by various types of DNases. And this is the protective mechanism of the cell. The DNases are responsible for digestion of the foreign DNA as soon as it enters inside the cell. It does not digest its own DNA, but whatever is foreign, it is immediately digested, and the DNA is not allowed to remain in the cell, to persist in the cell. So then how is it that the viral DNA manages to stay in the cell, to enter in the cell, to get protected from the DNases of the cell, and in spite of this, or a step ahead of this, how does the viral DNA conquers the cellular mechanisms of the host? How is it that the viral DNA, it, you can say, sends signals to the enzymes, to the protein molecules, to the ribosomes, to the DNA or the RNA polymerases for the process of transcription and translation of its gene? So, do you get my point? So, this is done, of course, because if it is not done, the DNA of the virus will not be able to produce the capsomers or the viral components. Many components are present depending upon the viruses. You can say the capsids, different types of capsids, different types of arrangements of capsid depending upon the architecture of the particular virus and the structure of the virus. We've seen the structure of, at the 12th standard, you must have seen the structure of T4 bacteriophage. So depending upon the variations in the structure, all the constituents of the capsid of the virus, it is synthesized, and this is synthesized in the host. So in case of T4 bacteriophage, you have not only have the ecohedral structure, you have the helical tube, a tail. Then you have you can say the tail fibers, and you have the collar and all the constituents which make up the complete bacteriophage. So all these major and minor constituents along with the DNA or the RNA, it has to be, you can say, replicated within the host cell. And the DNA or the genetic material of the virus, it does so by certain modifications, certain, you can say, tricks endowed by the nature um, to the viruses, by the virtue of which these viruses, they escape the defense mechanisms of the host, the DNases of the host, and they also manage to divert the macromolecules, the enzymes, the proteins, ribosomes, to its own DNA or the RNA, and leading to the transcription and the translation, which results in the production of the viral particles, and the assembly of the viral particles takes place. So this we will see shortly in the coming lectures how this is achieved by specific viruses. We have some viruses and its reproduction in your syllabus. So in the reproduction part, we will have the, you can say, uh, the details of the reproduction of the viruses. Now the viruses, they code efficiently. So there may be overlapping genes encoded within the genes. The small genome of the hepatitis B virus, which is a very good example of the overlapping and the genes encoded within the genes. So the genetic makeup of the viruses is responsible for the further translation and transcription and the translation of the viruses. And the coding of the genetic material, it is responsible for the, you can say, the lytic cycle or the lysogenic cycle of the viruses. So virus proteins which are produced from the viral genetic material after their decoding, after the transcription and translation, they are also found to be multifunctional. A single virus proteins may have several different activities or enzyme activities. So these are the parts of the, this is a, you can say, a concept where uh, related to the survival of the viral genetic material and how it plays a role in the eukaryotic or the prokaryotic cell, how it manages itself, manages the DNA, how it manages to conquer the cellular machinery and divert it towards its genome to produce several copies of its own for its survival. So the next is the viruses are parasites. So this point also I have mentioned it to you that viruses, I can say further that they are obligatory parasites because they have no survival outside the cell. They cannot, they do not have rather the requirements of the nutrition. And one more thing is that they do not have the nutritional requirements because as an independent entity, they are non-living. Independently outside the cell, they are not in the need of the metabolic activities, not in the need of energy, not in the need of any type of enzymatic cellular activities. So outside the cell, they are dormant in conditions, we can say. But their existence, their reproduction, it is dependent upon its attacking the various cells, which is a highly specific mechanism. It is not that any virus attaches to any cell and the infection begins. There are there is a high specificity in the types of viruses and the bacterial cells, which type of viruses attack which bacterial cells, as well as there's also a specificity between the different types of animal cells as well as the, you can say, the different animal viruses. So Just a minute. So, the viruses, they differ from the cells in which they, and the way in which they multiply. A cell, any cell, a plant cell, animal cell, a bacterial cell, it is always formed directly from the pre-existing cell. But a new virion is never formed from the pre-existing virion. Do you follow this? This is the major difference. A new virion, नवीन व्हायरल पार्टिकल किंवा नवीन व्हायरस हा आधीच्या व्हायरस पासून तयार झालेला आहे बॅक्टेरियल सेल सारखा किंवा कुठल्याही लिव्हिंग सेल सारखा असं कधीच होत नाही. So new virions are formed by a process of replication which takes place only inside the host of the host cells, and this involves the synthesis of components followed by the assembly into the virions. Therefore, we say that the viruses are parasites and they are dependent only and only on the host for most of their requirements. That is, as I told you, viruses, they do not have any activity of their own for their survival. The only thing is that the reproduction of the viruses, it is satisfied inside the living cells. And for the reproduction purposes, for increasing their numbers, viruses inside the cells, they are dependent on the host. So this point has already been seen, but on what components they are dependent? They are dependent on the building blocks such as amino acids, nucleosides, on the protein synthesizing machinery, and on the energy in the form of ATP or whatever the form that is required by the viruses. So this point we have seen, that is, a virus is capable of modifying the intracellular environment of the host in order to enhance the efficiency of the replication process. So the modifications, it includes the production of membranous structures, reduced expression of the cells or genes, or enhancement of the cell processes. So here, in case of photosynthetic bacteria and photosynthetic bacterial hosts, these also possess certain viruses, and when these viruses attack these photosynthetic bacterial hosts, one of the property is they are found to boost the photosynthetic activity of the host, and this may be a reason which enhances the multiplication of the viruses in case of photosynthetic bacterial hosts. So this is about the parasitic nature of the viruses. Now we will see about the ubiquitous nature of the viruses. So what do you mean by ubiquitous? Ubiquitous means they are present everywhere on the earth. There is no region on the earth where viruses are not present. Ubiquitous म्हणजे जळी स्थळी पाषाणी काष्टी सर्वत्र त्यांचं अस्तित्व, व्हायरसेसचं अस्तित्व आहेच आहे. So viruses infect all cellular life forms. That is, they infect the eukaryotic cells, eukaryotes rather, which include the vertebrates, the animals, vertebrate animals, invertebrate animals, plants, fungi, and the prokaryotic cells, which include bacteria and the archaebacteria. So archaebacteria is a different class of bacteria with unique properties. So archaebacteria are also found to be infected with the viruses. The viruses that infect the prokaryotes, as you all know, they are known as bacteriophages or phages. So the presence of these viruses, it is obvious in the host and they show some signs of the diseases. But there are many healthy organisms and which are infected with the viruses, and these viruses, they are non-pathogenic also. So it is not that all the viruses are pathogenic, such as bacterial cells, just like the bacterial cells. all the bacterial cells are not pathogenic is in nature. Similarly, viruses, though they are found to infect or they are found to enter inside the healthy organisms, healthy microorganisms, healthy human beings, healthy eukaryotic cells, it is not necessary that they will show the signs of the diseases. Some of the viruses are found to be non-pathogenic in nature. And even in these cells, when they enter inside the enter inside the healthy cells and do not show any signs of infections, it is not necessary that they will reproduce or they will be in an active phase of the growth. It may be that they remain quiescent. They are present in the cell, but they do not, you can say, enhance any of its activity, such as replication of the DNA or production DNA or RNA, or production of its of its capsid, or some activity of its own, which is decoded by the genetic material. So this infection, it may be active or the viruses may remain, you can say, quiescent. So further, the genomes of the many organisms, they contain remnant ancient virus genomes that had integrated into the host genomes long ago. So viruses, they are found to persist in the genomes of many in the cells, in the genomes of the cells of many organisms, where they have been integrated within the host genomes. And this integration, it is for generations together. And the viruses have not dislodged themselves from this integrations. They have not been separated out. They are inherited for generations together in this integrated form. So in this way, the viruses are present ubiquitously on the earth. So being present within their hosts, they are also found in soil, air, and water. Many aqueous environments also contain high concentration of viruses, and which are responsible for infecting the animals in those, or the organisms in those environments. So as we have seen that viruses are ubiquitous, they are also found in soil, air, water. Aquatic viruses are also found to infect different types of flora and fauna in the aquatic environments. Now, one more thing is that viruses, they are highly specific in nature. So only a few viruses and their life cycles have been, and their structures, of course, have been studied in detail. For example, the intestinal bacterium Escherichia coli. It has been a subject of study of replication of many types of viruses, because this has been considered as a standard organism in the scientific world. And therefore, all the viruses infecting Escherichia coli, specifically the coliphages as they are known, they have been studied in detail. So just see, this is just a part of it. And here it has to be, you can say, remembered that we have only a few examples which have been studied in detail, examples of viruses, where the structures of the viruses, the role of the viruses in the host, the reproduction strategy of the viruses in the host at molecular level has been studied. But here it has also to be, you can say, taken into consideration that as it is, microbial world itself, you can say, it has not been discovered to its fullest. This is one part. And whatever microorganisms are present, all the microorganisms, or you can say just 1 to 2% of the microorganisms have been studied for their hosts and their viruses. The existing microorganisms which have been discovered, which have been identified, have not been studied for the interactions between the viruses at the molecular levels. This is about the existing microorganisms, and it should also be remembered that there are microorganisms, many types of the bacteria, many types of, again, you can say the plants, which are yet to be discovered, yet to be isolated. And you can say there is again a class of bacteria which are known as the non-cultivable bacteria. There are bacteria which cannot be cultivated in the laboratory. So such bacteria which cannot be cultivated are also infected with the viruses. Here the point is, whatever we know about the viruses is just, you can say, a part. It is very little, and much more has to be dug out about the viruses. So we can say that the knowledge about the viruses or the infective agents, as it is, even the knowledge of microbiology, we will speak about here only about the viruses, the knowledge of these viruses uh, with respect to the structure of the different types of viruses, with respect to the infection of the bacterial cells, with respect to the changes that they make in the cells at the molecular level, whether they go for the lytic cycle, whether lysogeny, responsible for cancer production, responsible for the, you can say, storming the changes of the biochemical activities against the normal activities of the bacterial cells. All these aspects yet remain unstudied. So whatever has been studied, you can say this is just a tip of the iceberg. And you younger generations have much more to be discovered. So now we will see as to why we need to study the viruses. So naturally, you may have this question in your mind. Just let me confirm whether you are, you can say, online. Yes, can you hear me, students?
Yes, ma'am.
Yes,
Yes, ma'am.
there is no disconnection. Okay, no disconnection on the screen as well as on the voice. I am audible as well as you can see the screen. Okay, yes or no?
Yes, ma'am.
Okay, so we will continue further. So can you see it, please? Yes. So here we are seeing about the point, that is the reasons for studying the viruses. So naturally, this is a question that most of you must be having, that viruses as it is, they are smaller than bacteria. For the observation of bacteria, being students of microbiology, you you must be saying that as it is, observation of bacteria under the oil immersion lens is so difficult. And why we are interested in studying the viruses which are so many times smaller than the bacteria themselves? What if we just neglect them? So they are the part of the nature, and let them be. Why should be so inquisitive in studying the agents which cannot be seen, which have to be seen by the electron microscope? Okay, as far as the diseases are concerned, okay, we can consider some diseases which can be treated or which cannot be treated. But this is not the case. So the point here is reasons for studying the viruses. Here we will see why we are interested in studying the viruses. So the first point over here is some viruses, they cause disease. So here some, the word some, shabda jo aaye, the word some is important. All this itself says that all the viruses, they do not cause diseases. So some viruses, they cause diseases. And see, they are important agents of many human diseases ranging from the trivial, that is, for example, the common colds. Common cold, it is a very, you can say, old type of a disease in human beings, to the lethal, which is a rabies virus, rabies infections. Of course, we have certain, you can say, antidotes for rabies or rabies vaccines also. But if not treated, this this may be lethal, or proper doses if they are not given in case of rabies uh, dog bites. This yet today also it proves to be fatal. So this is one type. So they are responsible for many types of human diseases, ranging from common colds, which are a part of the, you can say, our lifestyle. And common colds are not lethal, but ranging from common colds to the lethal. Example is rabies and many types of infections. Next, viruses also play a role in development of several types of cancer, as we have seen in the oncogenesis. We have seen different types of viruses, and the hypothesis which explains the role of viruses in human cancers. So, individuals who suffer from the viral diseases, they are naturally responsible for the affecting the well-being of the society. So naturally, virally infected patients today, you know the scenario of the COVID-19 today. So this is a good example that this virus has affected the well-being of the society. आपण पहिल्यासारखं सोसायटी आपली जी आहे, आपण जी माणसं आहोत सोसायटी मधले, तर पहिल्यासारखं आपण वावरूच शकत नाही. सो दिस वेल बिईंगनेस, आधी कोविडच्या आधी आपला जसा सोसायटीमध्ये वावर होता, जो अतिशय मोकळेपणानं आपण सगळ्या गोष्टी उपभोगत होतो, त्याच्यामध्ये त्याच्यावरती कितीतरी बंधनं तुम्हाला आलेली दिसतात. आणि आज आपण जे काही जगतो आहोत, दिस कॅनॉट बी सीन as the well-being of the society. So virus diseases, they affect the well-being of the society. Today's disease COVID-19 is a good example of the threat that is being caused by the virus in the society. Threat of to be or not to be, threat of death of the individuals infected by this disease. So this is definitely not the well-being of the society. So the example again, in the past we can see smallpox had a great impact. Now the smallpox has been, you can say, eradicated. And in the newer cases, AIDS is also one of the disease spread by the viruses, and several other diseases also, which have made a great impact. The SARS viruses, the MERS viruses, they also have a great impact in nowadays, or in the coming decades. In coming 10 years, these viruses have changed the scenario of living of individuals to and far, in India as well as over the globe. So over here, I have a picture of viral diseases in front of you. So we are dealing with the point of viral diseases. So these are the viruses that infect the human beings. This is a smallpox. You can see this is disease caused by the smallpox virus. Okay, so the virus is harmful, and the harmful effects of the viruses. In vertebrates, foot and mouth diseases. Okay, foot and mouth diseases in case of the vertebrates. In case of invertebrates, so the leather jackets infected with tubula iridescent virus. So this is an invertebrate which is infected with the viruses. In plants, a very good example is you can say, a damaged potato, okay, with the viral diseases. So the spraining of the potato caused by the viruses. And again over here, the infection of the potato plants by the viruses, they are responsible for delayed emergence of potato caused by the retrovirus infection. So this is, even if there is delayed emergence of potato, this results in the, you can say, infection of the potato by the virus and certain, you can say, discoloration, and the cells of the potatoes, they are, uh, you can say, damaged by the viruses. So this is about the viral infection in the plants. Next is the fungi. Mushroom viruses are also found, and in case of bacteria, so this is a Escherichia coli cell, which is attached with the T4 bacteriophage. Okay, so here we were seeing the point of the harmful aspect of the viruses, that is disease causing viruses. There are also some beneficial aspects of the viruses as to why we have to study the viruses. So one is the different, because they have different types of diseases, or they are responsible for causing different types of diseases. Therefore, uh, as they are responsible for causing much harm to the human being specifically and to the society. Therefore, it becomes a requirement to understand the nature of the viruses, how they replicate, how they cause the disease. So, this knowledge of the नेचर ऑफ द व्हायरसेस अँड देअर रेप्लिकेशन का बघायचं आहे, कारण हे तुम्ही ज्या वेळेला अभ्यासाल, when you have this knowledge of the nature of the virus, the structure of the virus, infectivity of the virus, how it infects the cells, human beings, which cell is attached or attacked by the viruses, how they replicate, how they cause the disease. This knowledge, it permits the development of effective means for prevention, diagnosis, treatment of the virus diseases through the production of vaccines, diagnostic reagents and techniques, and antiviral drugs. These medical applications therefore constitute a major aspect of science and virology. आताची कंडिशन ही अशीच आहे, तंतोतंत आपल्याला व्हायरसच्या स्टडीज या आस्पेक्टला आपल्याला अतिशय दुजोरा देणारी आहे. कोविड-19 असा व्हायरस आहे की ज्याच्याबद्दल अजून फारसं माहिती नाही, अभ्यास चालू आहे, पण त्याचं डिटेल नेचर ते कधी बदलेल हे सांगता येत नाही. एक तर स्ट्रक्चर जरी तुम्ही आत्ता म्हणालात स्ट्रक्चरल आस्पेक्ट त्याचा आहे, तर ठीक आहे, स्ट्रक्चरल आस्पेक्ट्स वरती आता या मुद्द्यापुरतं बोलायचं झालं तर आज कोविड-19 मध्ये आपण स्ट्रक्चरल आस्पेक्ट स्टडी करणं गरजेचं आहे, त्याचं रेप्लिकेशन कसं आहे करणं गरजेचं आहे, त्याचा यू कॅन से डिझीज प्रोडक्शन कसं होतं, कोणते कोणते सिम्पटम्स दिसू शकतात. The variety of symptoms are seen today in this COVID condition. फक्त you can say difficulty in breathing, lung infection एवढाच एक aspect नाही आहे, प्रत्येक वेळेला नवीन नवीन पिक्चर्स तुमच्या समोर येतात. तर अशा कंडिशन मध्ये ज्या वेळेला कोविड हा पॅन्डेमिक झालेला आहे, ग्लोबली इन्फेक्टेड आहे, सगळं जग ज्यांनी इन्फेक्टेड आहे, सगळ्या जगामध्ये ज्यांनी हॅवॉक क्रिएट केलेला आहे, इन्फेक्टिव्हिटी रेट जास्ती आहे, डेथ रेट जास्ती आहे, आणि त्याच्या विरुद्ध कोणत्याही प्रकारचं औषध नाही. वी डू नॉट हॅव अँटीव्हायरल ड्रग्स which target the COVID-19 exactly. Okay, we have generalized done. आताचे जे ड्रग्स वापरले जातात ते जनरल ड्रग्स आहेत, पण आपल्याकडे असा कोविड-19 साठी कोणत्याही प्रकारची यू कॅन से अँटीव्हायरल ड्रगची ट्रीटमेंट नाही. त्याचं डायग्नोसिस कसं करायचं, डायग्नोसिस हे परफेक्ट कसं असणार आहे, कोणत्याही प्रकारची चूक न होता तुम्हाला हे सांगता यायला पाहिजे की कोविड पेशंट आहे का नाही, त्याचे टेक्निक्स डेव्हलप करणं, प्रोडक्शन ऑफ वॅक्सिन्स which will result in giving immunity to the public as a whole. So such type of studies are required and they are being done widely in the science of virology. So this is a very important aspect in today's condition. Why virology? Why we need to study the viruses, and what is the, we can say, the broad perspective of virology. So this is a very vast field, and today's scenario, it is a very, you can say, applicable field, an emerging field, which the discovery in this field, which will be a boon to the human society with respect to COVID-19. So in veterinary virology, plant virology, these are also some of the important branches which have an economic impact on the animals as well as the plant. So many viruses cause diseases in the domestic animals, and also viruses responsible for crop plants. So we have seen about the foot and mouth diseases, the rice yellow mottle viruses. These are the two examples which represent the seriousness of the viral diseases in the plant as well as the animals. So such type of infections, they are responsible for causing economic damages in the dairy industry, and for example, where the phages are found to infect the lactic acid bacteria. In such cases where the lactic acid bacteria are infected with the phages, they are responsible for fermentation losses that are used to produce the cheese, yogurt, lactic acid, etc. So this is the, I think this is all for today. Okay. So again, the student number is just 43. Okay. Okay. So just have a conversation with me and leave your chats in your chat box. Yes, all of you. Be very fast because you are about to start with the next lecture. Not interested in having any conversations?
हॅलो मॅम. मॅम सगळं समजलंय, फक्त फर्स्ट जी स्लाइड दाखवली ना त्यातला सेकंड पॅराग्राफ नाही समजला.
फर्स्टची स्लाइडच्या सेकंड पॅराग्राफ, बर ती स्लाइड कोणती होती ते जरा बघून ठेवा. पुढच्या लेक्चरच्या वेळेला तो तुम्हाला समजून सांगितला जाईल. ओके
ओके मॅडम.
ओके. कारण का आता दुसऱ्या मॅडम असतील तर दुसऱ्या मॅडम जॉईन होतील, मॅडम किंवा सर असतील आपले, तर ते जॉईन होतील. तर आपला वेळ संपत आला ना, बरोबर आहे. येस, 12:15 पर्यंतच आपला वेळ आहे. तर ऍक्च्युली आपण बराच वेळ एक्सटेंड केलेला आहे लेक्चर. सो आय थिंक आय शुड लीव नाव. ओके. येस. ओके. So in the next lecture, we will be studying about the further aspects of the viruses. And here we have seen about the viral diseases. We will see about the, you can say, the beneficial aspects of the studies of viruses. Okay. So, okay. I will be leaving students. Stay home, stay safe.